As you can tell, it takes a scientific village, and it's just wonderful to see the progression of all of these projects, not just Dr. Heafy's, but a lot of the work that we've been happy enough to support over the last few years. So after years of science, if all goes well, then some of these discoveries make their way into a clinical trial. And clinical trials are the opportunity for you as patients to become involved, and we'll hear more about that. Our next speaker will talk about this. Dr. Hussein Assi is an assistant professor of medicine at Boston University School of Medicine and a medical oncologist at Boston Medical Center. His work focuses on novel therapeutics in NETs.
I want to thank everyone for being here today and NETRF for inviting me to give this talk about clinical trials. All right, so these are my disclosures. So outline. So today I'm going to talk about, I'll start with the principles of clinical trials, and then I'll follow with clinical trials specifically in neuroendocrine tumors. And I'm going to end with, with information about where you can find clinical trials within your area. So this is a slide that shows the advancement in neuroendocrine tumors treatment throughout the years. So as you can see, since the 1980s through like 1990, there were 2 treatments. But then starting in 2011, there's been an explosion in terms of the number of treatments in neuroendocrine tumors. And all these happen because of results from clinical trials. So what are clinical trials? So they are a type of research studies in which people volunteer to help find answers to specific health questions. So when we do them carefully, they are the safest way to find new treatments and ways to improve health and patient care. So when we do health clinical trials, we do them based on what we call a protocol. So that protocol is a document, and in it, it has lots of information about what patients are involved, so the type of patients with certain criteria, the schedules of the tests and procedures that are to be done throughout the lifetime of the clinical trial, the drugs involved and are to be given and how they are given, the length of the study and how long the patients will be followed up for, and the exact objectives of the study. What are the questions that we're hoping to answer in those clinical trials? So why do we need them? I listed here like 3 main reasons why we need clinical trials, especially the treatment clinical trials. So the first one is really to determine whether a new drug or device is safe or effective. Another reason would be to study different ways where current standard treatments may be used more effectively or safely, or how to sequence certain treatments that we already have approved. A third reason would be to assess the effectiveness and the safety of an approved treatment in a population that was not previously studied in previous clinical trials. So here I included some truths and myths about clinical trials. So let's start with the truths. So whenever you decide to participate in a clinical trial, it is totally optional, and you can change your mind at any time. That means you can withdraw your consent if you feel that, if you have concerns about the clinical trial, or you found that there's another treatment or another clinical trial you'll be interested in. Really, for whatever reason, I just wanted to make sure that you understand that clinical trials are not something that is imposed on you. It's just something that you participate at will. Another one is that you cannot directly compare results of one clinical trial to another. And the reason we cannot do that is because each clinical trial look at certain patients with certain criteria. And these criteria can be different from one trial to another. So even though sometimes you may seem inclined to just look at one clinical trial and look at the result and compare it to another and say, look, this one is better than this one, it's very important to look at the details of what are the patient population that has been looked at in those trials. The third truth would be that in terms of cost. So usually companies or the clinical trial that are funded by pharmaceutical companies or funded by the NCI, the cost of the drug is provided by them. However, there are certain costs that are related to standard of care procedures, and those just are billed regularly through your insurance. Now, myths about clinical trials. So clinical trials are proven better than standard treatment. So this is not true. So, and the reason is because the clinical trial is there to look as to whether the treatment is better than standard of care. We don't know that, and that's what the clinical trial is trying to answer. A second myth would be the clinical trials are for patients without other options. So while that may be true in some cases, there are lots of clinical trials where patients who have treatment options decide to join a clinical trial to look at a potential new drug that is being studied, even though there are other drugs that they could have taken as a standard of care. So it's a spectrum. And the third myth that I want to talk about is, so you will unknowingly get a sugar pill or a placebo. So a placebo is basically, it's like a sugar pill. That means you're being given something that we know has no effect. And we do that usually so that we can compare a new drug to that placebo to see if there's an actual effect. So usually, while that may be true, there are some studies where you can be on a placebo. Usually it's very clear that you can be on a placebo, you'll be randomized, and I'll talk about randomization in my next slide, to the placebo. First of all, it can be blinded, so that's true. You can be put on a placebo without actually knowing that you're taking the placebo. It will be very defined at the beginning of the protocol. What are the chances that you'll be on that placebo? Okay, now in terms of clinical trials and who gets on clinical trials. So overall, based on some studies, it's estimated about 7.1% of cancer patients participate in treatment clinical trials. So this number is pretty low when you look at it, but it varies. And it varies on various factors. So you have, it varies by type of the cancer center. So as you can see here, in bigger NCI-designated cancer center, this number can go up to 21.6%, while in some community programs, it can be around 4.1%. It also varies by race and ethnicity. So there's a study published a couple years ago that looked at that. As you can see here in the numbers, the average was 7.2% among whites, 4.4% among blacks, and 4.2% among Latinos. So you can see here there's some discrepancy in there. So what are the phases of clinical trials? So, and this is important because each phase tries to answer a different question. So first, we start by the preclinical phase. So, as Dr. Heafy mentioned earlier, there are models that we look at prior to looking at drugs in humans, and those are cell lines, animal models, like mouse models. And this is where we gather basic information about the safety and biological efficacy of a certain product. And then once we have that and we have some signal that something is potentially beneficial for in a certain type of tumor, then we go on to the phase 1 part of a trial. So a phase 1 clinical trial tries to focus on the safety and determining the proper dose in humans. And usually these are small studies, 15 to 25 patients, and usually they're across different tumor types. Then phase 2 studies, so after a drug makes it through a phase 1, it goes through a phase 2 study. A phase 2 clinical trial looks at the preliminary test of efficacy and also side effects of this drug in question. And usually this is about 25 to 50 patients, and it's within a specific tumor type. Once the drug shows potential efficacy in a phase 2 trial and it's safe, then it goes on to phase 3. So the main aim of phase 3 trial is to really know whether the drug is better than what we already have. So it compares a new treatment to a standard treatment. And usually these are bigger trials. We're talking about 200 to 500 patients, it can be even more sometimes. And this is really the gold standard. This is where a drug shows clear efficacy over a standard, and that's where we go to the FDA and say, listen, we have this drug, we have phase 3 data, we want it FDA approved. And then we have the phase 4, and this usually happens after FDA approval, and this is really just to test the long-term safety of drugs. And the reason we do that is because during the phase 3, this is a study that usually takes 2 years, 3 years, 4 years maybe. So we don't know the long-term implication of whatever new treatment that we studied in phase 3. And it's very important that we keep on looking in the long run to see if there are any signals or any safety signals or side effects that we may have missed during those first few years during the patients, during the phase 3 portion of the clinical trial. All right, so in terms of who sponsors those clinical trials, where do they come from? So there are 3 types here that I listed. So there's investigator-initiated trials, there's industry-sponsored trials, and there's trials through the National Clinical Trials Network. So as you can see here, the investigation-initiated trial, as the name sounds, it's initiated by an investigator. So they're usually smaller trials, phase 1, phase 2. They're initiated by an academic MD, but they're funded by either the institution, philanthropy, or pharmaceutical company. Industry-sponsored trials are funded by pharmaceuticals, and the idea is also initiated by the pharmaceutical company. They can be phase 1, 2, or 3, and usually these involve many sites and can be international as well. And the National Clinical Trials Network also involves phase 1 to phase 3 trials, and they're initiated by what we call cooperative groups, such as ECOG, SWOG, Alliance, and NRG. So this is funded by the the NIH and the National Cancer Institute, which funds these 4 different cooperative groups. So this is just to kind of just show the life cycle of a drug. So after the basic science of the drug discovery and the preclinical data that shows potential biological effect of a new drug in a tumor or in a tumor cell, then you apply for an investigational new drug or an IND, which is submitted to the FDA before initiation of a clinical trial, so before you try the drug in a human population. After the drug goes through the phase 1, 2, and 3, that's when you can apply for the new drug application, or NDA, which is submitted after the phase 3 is complete, and then the FDA reviews the data, all the data from the beginning all the way to the phase 3, and that's when they issue FDA approval. And this is really the finish line for a drug. And after that, you have the post-market safety monitoring, like I said, to monitor for any safety signals, long-term safety signal, which is the phase 4, basically. Now, within the different phases, there are just different trial designs, which I wanted to briefly highlight. So for phase 1, usually there are 2 types that I want to mention. So there's dose escalation, and as the name sounds, you're basically starting with a low dose of a certain drug, and then you're going up to see what's a safe drug, what's the maximal tolerated dose that you can give a patient. After you determine that, typically the drug goes through a dose expansion, where once you determine that dose, you try this drug in a certain subset of population with a certain tumor or same disease. Once you've determined the safety and preliminary efficacy in a phase 1, then you go to a single-arm phase 2, which is you give the drug to a larger patient population with the same disease, but you're not really comparing to anything right now. You're comparing the results to results from other trials that have been done in the past. So this is what we call historical control. So this is not enough yet. That's what we need to do phase 3 trial, and sometimes phase 2 can be randomized. And I'll explain what randomization is, but that's when you really compare an experimental treatment to another arm, which is the control treatment. And the control treatment can be a standard of care that you already have, that's already been approved, or it could be a placebo. And usually it's 2 arms, so experimental control, but sometimes it could be more than 2 arms. You can have like 2 experimental arm and 1 control arm. And this is really the gold standard that is used to get FDA approval. So as promised, randomization. So we need methods to really minimize our bias and maximize the validity of the results. So randomization is basically dividing patients by chance into separate groups so that you can compare the treatment and the control. And the reason you want to randomize is you want to make the 2 groups as similar as possible before you give them the treatment and the control. Because you want to make sure that it's the treatment that's doing the effect that you're noticing or that you're evaluating for, and not something else. So it's very important we try to achieve, as much as possible, similar groups in those 2 arms. Blinding is when the study participant, clinician, and/or outcome assessor do not know which intervention is received. And we do that because we want to try to decrease the bias. It's because we know, as humans, if we know that a patient, or if the patient knows that they're getting a treatment, they're more likely to feel that that they're getting better. But this may not be true. So we want to minimize that, and that's why blinding is sometimes used in studies. So in terms of design and interpretation of clinical trials, so whenever you design a trial, like I said, you have certain criteria that are needed to include those patients in a trial. So in neuroendocrine tumors, for example, what is the grade that we're talking about that is being evaluated? What is the stage? What primary side are we talking about? Pancreatic neuroendocrine tumor, a GI? Is it growing or not in the past 12 months? Is this a first-line treatment, second line, third line? And what we measure really is these are some of the measurement of success. I just listed some of them that are commonly used. The response rate, which is how much the tumor shrinks. The progression-free survival, which means how long does it take for the tumor to grow back? Overall survival, which is, as it sounds, is just how much patients survive total. And then the safety of the drug. And this is just a study schema that just shows how a trial protocol is. You can see here, you have the criteria. They're randomized into 2 arms. This is treatment arm, control arm, and these are the endpoints over here. So this is an example of the NETRF trial that was mentioned before. As you can see here, this is the progression-free survival. So it shows that the lutetium dotatate, the PRRT, has a higher PFS than the control arm, 65 versus 10.8. And as you can see here, a lot of time you'll see hazard ratio and a p-value. So what these means, the p-value is just it tells you how confident we are that the treatment, the effect that we're seeing is actually due to the treatment and not to chance. And we want this typically to be less than 0.05. That's the magic number that we usually look at. And the hazard ratio, we want it to be less than 1, which tells us that the treatment is actually better. When it's 1, that means it's no better. If it's more than 1, it means it's worse. So the lower the number, the better. And these are safety endpoints that we look at. So adverse events, so we grade them. Grade 1 is mild, moderate, severe, life-threatening, and death. So that's how we grade adverse events or side effects, basically. And then it's very important also to know whether if the investigator thinks that the adverse event is unrelated, likely, unlikely, possible, probable, or definite. So this is just an example of how we grade the different side effects, and this is based on the common terminology criteria for adverse event, CTCAE, which we use often. This is just in the table format how it looks like. So we have the grading. We look a lot at grade 3 and grade 4 because those are the most prominent, and those are the ones that where we need to intervene and reduce the dose. Here I listed some of the key ongoing neuroendocrine tumor trials that are currently open to enrollment. As you can see here, you have the ACTION-1 trial, for example, which looks at an alpha emitter, RAISE 101 versus standard of care. You have here another alliance trial of INPARA and FAO, paraganglioma and pheochromocytoma that looks at temozolomide plus or minus olaparib. You have here the COMPOSE trial, which is a phase 3 trial looking at dotatate versus standard of care. And retreat that was also mentioned earlier, which is retreating with Lutathera. So we're hoping to really derive data from these that will help patients in the future as to whether these interventions are effective or more effective than what we currently have. And these are some more trials as well that I mentioned here, some in lung, some are, these are adjuvant. So this one is a randomized phase 2 in adjuvant pancreatic neuroendocrine tumor. That means patients are treated and we want to make sure, or want to try to decrease as much as possible the chance of recurrence. And how you can learn about clinical trials. So your oncologist and neuroendocrine tumor doctor is really your primary guide here. But if you go to your institution's website, usually there's a website, there's a site, there's a place where they list clinical trials that are available at those centers. And then these are some other sources. Clinicaltrials.gov is a main one. So this is right here. You can type in your tumor type. You can type in stage, if the trials that are open, and then by location as well. And you can get a list of clinical trials. And same thing on the National Cancer Institute site. And then, so the take-home points are basically that clinical trials are needed to advance the field and coming up with new treatments for neuroendocrine tumors. And this has been going on very rapidly in neuroendocrine tumors, as you can see, over the past 13 years. And hopefully in the future, radioligand therapy, targeted therapy, liver-directed therapy, and personalized therapies are likely to be incorporated in future NET trials. And with that, I end my presentation. Thank you so much.